Hello and good morning from Adelaide. Today you'll have to use your imagination. Imagine that you are holding a glass of a lovely Viognier wine. You swirl the glass, smell the aroma. Delicious apricot. But why does this glass of wine smell like apricots? To try and answer that question, we investigated wines with varietal stone fruit characters. So think about yellow and white peaches or nectarines and apricots. They could be fresh, cooked or dried. Stone fruit aromas are recognised by winemakers, wine judges and wine writers as desirable and important to a number of white wine varieties, such as these shown here. For the wines, for this study, we focused on Chardonnay as the major Australian white wine variety, Viognier because of its often evident apricot character, as well as botrytised Semillon. For some background, so everyone is on the same page, wine flavour mostly comes from the aroma compounds in wine. These are the compounds that you can smell when you sniff the wine in the glass. These volatile aroma compounds can come directly from the grapes, or they can be made by the yeast or released during fermentation. Then after that, they can come directly from the barrel, or they can develop during barrel or bottle aging. There could be hundreds of different aroma compounds in any one glass of wine. They include many chemical families such as esters and alcohols across a wide concentration range of grams per litre to nanograms per litre. So again, with your imagination, imagine one small drop of compound added into an Olympic-sized swimming pool. That would be nanograms per litre or parts per trillion. Impact aroma compounds are compounds that individually cause the one main aroma character. Some impact aroma compounds found in fruits and vegetables have also been found to be the impact aroma compounds to give the same aroma attributes in wines. For example, isobutyl methoxypyrazine, IBMP, for capsicum in Cabernet Sauvignon, 3-mercaptohexanol for passion fruit in Sauvignon Blanc, and rotundone for black pepper in cool climate Shiraz. So, could stone fruit flavour in wine be related to flavour compounds already known in actual stone fruit, or is it from unknown flavour compounds? In fresh apricots and other yellow stone fruit, the gamma and delta lactones are considered to be impact aroma compounds in that they smell like stone fruit. Other wine aroma studies have suggested that they were the cause of peach or apricot aromas in wines, but a previous study on lactones in wine, done at the AWRI, found extremely low concentrations of the gamma-lactones in only a few wines. So we thought it was unlikely that they would affect wine aroma. Also here listed are many other aroma compound families that are also often found in wines. But the question remained, which aroma compounds causes the stone fruit aroma in wine? To start answering that question, we set about deconstructing wines with stone fruit attributes. I should let you know, sometimes when I first meet people and the conversation gets around to where do you work, and I tell them the Australian Wine Research Institute, they often get a smirk on their face or giggle because they think that I'm joking, but I assure them it's a real place. This is then followed by the question, do you just drink wine all day? Well, no, but one of the perks of my job is tasting a lot of wines, and this was definitely the case for this project, because it was essential to have an appropriate samples for the studies. Over 100 Chardonnay, Viognier or botrytised Semillon wines were pre-selected because of their back label descriptions, winery tasting notes or suggestions from colleagues. The wines were assessed by small panels of expert wine tasters, up to 12 wines at a time across 10 sessions. From these tastings, sets of wines covering a wide range of stone fruit descriptors and intensities were chosen for further sensory studies. A good place to start this type of project is with gas chromatography, mass spectrometry, olfactometry, or GC-sniff investigation. A set of 10 wines, Chardonnay, Viognier and botrytised Semillon, that was 5 with high and 5 with low levels of stone fruit aroma, were assessed by GCO using an experienced panel of 4 people. In the GC, hundreds of wine aroma compounds separate into discrete peaks, hopefully, and are sniffed as they come out of the other end of the GC, while at the same time being detected by a mass spectrometer. Unfortunately, but not completely unexpectedly, no stone fruit aroma zones were consistently sniffed across all of the high stone fruit wines. The overall aroma profiles of the wines by GCMSO were similar, but had differing levels of some compounds. Viognier wines were relatively higher in monoterpenes. The monoterpenes linalool, alpha-terpineol, geraniol were described as floral or citrus. Some fruity, lolly-like esters were at higher levels in the high stone fruit Chardonnay wines. Interestingly though, gamma-nonylactone was detected as peach in the botrytis Semillon samples. So it could be that stone fruit in different wine varieties is caused by different compounds, and or a mixture of aroma compounds is required. Then, to try a different tack, I was fortunate to be able to spend nearly 3 months at the ISVV in the University of Bordeaux with Professor Philippe Darrieux's research group to try a trace analysis technique that they have successfully used to help identify several wine aromas. The protocol is quite straightforward, but it's an interesting mix of separation methods and involves some old school chemistry with some new school chemistry and lots of GC-sniff. Several French Viognier wines with definite apricot aroma were selected for the study. Almost an entire bottle of wine is extracted with dichloromethane. Combined phases are concentrated down to 1 ml. Then using HPLC, a quarter of the crude extract, that equates to roughly a quarter of a bottle, is separated into 50 fractions. These fractions are in ethanol water. Therefore, they can be smelled directly from the vial or after being poured into a wine glass. Some fractions smelled like nail polish remover, others vinegar, confectionery, pepper, oak, caramel, banana, etc., etc. But most importantly, apricot was detected in 3 fractions by a sensory panel. We were very excited by this outcome. So then it was back to the GC-sniff. But unfortunately here as well, no stone fruit or apricot aromas were consistently detected by GCO. Identifying the main aromas in the fractions by GCMS were some esters with fruity aromas, plus the monoterpenes linalool, alpha-terpineol and geraniol with citrus, pine and floral aromas, and the yummy, jammy beta-damascenone. So again, it was looking like a combination of compounds was responsible for apricot aroma in Viognier wines rather than one impact aroma compound. Not 100% successful, but it added to the story and backs up the original GCO. That work was followed by a very comprehensive study with a full descriptive sensory analysis, 6 Chardonnay wines and 6 Viognier wines from Australia and 6 Viognier wines from France, with a broad range of stone fruit intensities were used. The sensory panel provided 17 aroma descriptions plus palate terms. Apricot was considered reminiscent of preserved or canned cooked apricots, and peach was more like fresh white peach. Shown here are the results for 4 of the most different wines. Varied ratings can be seen for apricot and peach. And the 2 attributes were not closely correlated. So we did have a really good sample set of wines. As an added extra, another descriptive sensory study was conducted on this same selection of wines in Bordeaux at the ISVV. Interestingly, peach is not a term commonly used to describe Chardonnay in France. Even so, the apricot intensity ratings for the wines by the ISVV sensory panel were very similar to those found with the AWRI sensory panel. The sensory analysis was followed by a comprehensive quantitative chemical analysis. Over 100 known wine aroma compounds were accurately measured based on published stable isotope dilution analyses plus standard wine analyses. The sensory attribute ratings for all of the wines and all of the chemical results were correlated using Unscrambler X statistic software, and this came out. It might look very complicated, sorry, but what it shows is that there is more of these aroma compounds, the blue dots, in the wines that have stronger apricot or peach aromas. That is, those aromas plotted near the aroma attribute are predicted to be important for the wine to have those aromas. Looking at those important compounds in more detail, the peach aroma attribute was associated with acetate esters and monoterpenes, linalool, geraniol, and nerol. For apricot, gamma-lactone, gamma-decalactone, and the previously little studied and potent dairy lactone were all at very low concentrations. The monoterpenes, linalool, geraniol, and nerol smell floral and citrus-like, but were quite high in the apricot wines, higher than you might find in a floral variety like Riesling. And the C6 compounds, E2-hexenal and E2-hexenol, plus other aldehydes, non-enal and benzaldehyde, were at varied concentrations and have a range of descriptors. This trio of oak compounds down here were probably influenced by the French Viogniers. If we go back to the slide with the fresh apricots and other Yellowstone fruit showing the gamma and delta lactones, with the most important being the gamma decalactone, but also noted are some aldehydes and some monoterpenes. These compounds are starting to sound familiar. In apricots, gamma decalactone is often around 500 micrograms per kilo, but in the wines with high levels of apricot aroma, the amount of the gamma lactones found were quite low, only around 1 microgram per litre of gamma decalactone, not 500. Also, if you compare the low concentration of gamma lactones found in the wines, shown here, to the aroma thresholds that have been found, you would see that it's apparent that the individual gamma lactones were not major contributors to the apricot aroma in the wines. But there has been some previous research done in Spain that showed an additive or an even synergistic effect when lactones are combined. So when mixed together, they are perceived with a stronger aroma. So where to from there? Reconstruction. So the apricot and peach aromas in wine have been deconstructed, and so then we needed to reconstruct them. Reconstitution sensory studies were needed to confirm the importance of these compounds to the peach and the apricot aromas in wine. However, in the preliminary bench tastings, the peach reconstitution was not very convincing. Therefore, we only went ahead with the apricot. A control model wine was prepared, including a range of non-volatile compounds, plus 55 common aroma compounds at their regular concentrations, the average that they were across the 18 wines. This one was the control. These ones had the 3 lactones added on top, or 3 monoterpenes, or the 4 aldehydes added. And then there were all the other combinations. An aroma-only descriptive study was conducted, and the panel again selected apricot as one of the 13 aroma descriptors. The control model was rated low in apricot. No difference was rated for the lactone addition or the aldehyde addition. But with the monoterpenes added, the apricot aroma was rated significantly higher than the control model. In fact, all models with monoterpenes added were all more highly rated for apricot than the control model, confirming that the monoterpenes linalool, geraniol and nerol must be present in a glass of dry white wine in the correct ratio and each at the right concentration for it to have an apricot character. Another very recent Reconstitution sensory study, looking more closely at the interaction of the monoterpenes with the aldehydes and the gamma-lactones, confirmed once again the importance of the monoterpenes for apricot aroma in wine. And it's always good to do things twice. So the key findings from this project so far are, in the detailed sensory study of Chardonnay and Viognier wines, apricot and peach were able to be differentiated as two separate and distinct attributes. Apricot was considered reminiscent of preserved, canned or cooked apricots, and peach was more like fresh white peach. Comparing the sensory results to extensive chemical analyses of the wines, more than a dozen aroma compounds were found to be associated with the apricot and peach attributes, including gamma-lactones, monoterpenes, aldehydes and acetate esters. In a pivotal step, Reconstitution sensory studies confirm that the mixture of 3 monoterpenes, linalool, geraniol and nerol was the most important group to be added for the model to be perceived as having an apricot attribute. This result was surprising because the monoterpenes linalool, geraniol and nerol are usually described as floral and citrus-like. The levels of monoterpenes were quite high in the wines with more intense apricot aroma, higher than you might find in a floral variety like Riesling. Even though the gamma-lactones are the most important aroma compounds in actual stone fruit, this result in the wines negated the assumption that gamma-lactones were responsible for stone fruit aromas in dry white wine. And this was the first documented application of a comprehensive approach involving sensory, chemical, statistical analyses and reconstitution experiments, using wines explicitly selected with that attribute, that is, apricot. And just a bit more on the monoterpenes, and they're grape-derived aroma compounds. Monoterpenes exhibit floral, citrus aromas, and their aromas, as we've discussed, are also thought to be additive. And there's nearly around 70 compounds have been, or 70 monoterpene compounds that have been identified in grapes and wine so far. The most common aromatic ones found in some varieties of wine are geraniol, linalool, and alpha-topinol, as well as nerol, citronellol, and cis-rose oxide. In grapes, the monoterpenes can be found bound to 1 or 2 sugars. Here's an example of some chemical structures, and here is a stylized version. In the bound form, they are not volatile, and therefore they have no aroma. In other varieties like Riesling, Gewürztraminer, and Muscat, the bound forms are much more prevalent in the grapes than the free form, and mostly found in the geraniol glucoside as geraniol glucoside. During fermentation, some of the bound monoterpenes are hydrolyzed or broken apart by the acid during fermentation, releasing the free monoterpenes. Then the varietal aroma can be smelled. Some of the glycosides, though, or the bound monoterpenes, remain in the wine. Once the monoterpenes such as geraniol are freed, they can rearrange into other monoterpenes like linalool and alpha-topinol. I won't go into that complicated chemistry today, but my colleague Josh Hickson talked about it in his recent webinar on aged characters in Riesling, and another colleague, Mango Parker, will be discussing in-mouth release of monoterpenes in her webinar next month. Other research on other varieties have found that the monoterpene levels drop quite quickly over the first 2 years, but we have found some Viognier wines have maintained their high levels for longer periods, and their apricot aroma as well. A formal stability trial in Viognier wine is needed to confirm this. This new finding of relatively high levels of monoterpenes in Viognier wines led to vineyard studies. Samples were taken of 11 Viognier clones immediately prior to harvest from a vineyard in Eden Valley and stored in the freezer until analysis. The main monoterpenes, linalool, geraniol, and alpha-terpineol, nerol, etc., were measured, and all had a very similar profile between clones and between vintages to that seen here for linalool. Although in 2015, Yalumba 1 was harvested early. It was a commercial decision, and therefore there's no sample that time. So there are vintage differences as well as clonal differences. In a further study of 4 popular Viognier clones, 2 clones showed similar monoterpene concentration profiles throughout ripening, but both other clones were substantially lower in monoterpene concentration. This presents an option of clonal selection as a tool to modulate apricot aromas in Viognier wines. We are also measuring the bound monoterpenes in all the clonal samples and the ripening study samples. This is ongoing, so I cannot show you any results yet, but stay tuned for results at the next Australian Wine Industry Conference. Until this project, Viognier had been little studied. This research has contributed new knowledge of both wine and grape composition for this variety and clarified its classification as an aromatic variety. So to continue on with this work, we'll be doing some more reconstitution studies. They're scheduled in the next months to look at the monoterpenes one at a time and also to see what is really needed within the background 55 compounds. Also, by checking the effect of adding the important compounds, especially monoterpenes, into a neutral white wine. Then perhaps investigating peach in Chardonnay wines more specifically, finalising the bound monoterpene measurements and comparing those results to the free monoterpenes across the clones and ripening time points. And then we can see what that tells us. Also, from the results of the ripening study, a winemaking study is planned for this coming vintage at 2 sites with 2 clones at 2 ripening time points to investigate how compositional differences in Viognier grapes relate to wine composition and sensory properties. I didn't talk about dairy lactone much today, but is 100 times more potent than gamma-decalactone. It is down in that nanogram per litre level that I was talking about early on. From a survey of over 100 wines from across Australia, more was found in Riesling and Viognier wines than Sauvignon Blanc, Chardonnay and Pinot G, but still not very much. This compound, though, is more likely to be of importance in the botrytised wines and possibly red wines. Therefore, surveys of these wines will be conducted. Also looking at the different ratios between the monoterpenes and other wine varieties that are often described as having an apricot attribute. So thank you for joining in for this webinar today. We believe that research to understand these flavours is highly valuable to the wine industry. Regarding the importance of monoterpenes to the distinct apricot aroma of Viognier wine, information on the differing levels of monoterpenes between Viognier clones and the optimal level of monoterpenes in the grape could aid grape growers and winemakers in their decision of when to harvest or which clones to plant. Also, due to the high monoterpene levels identified, Viognier could become more commonly used by winemakers as a blending option to obtain desired wine flavours. Many thanks to my co-authors. I would also like to thank my PhD supervisors, as this project was the basis of my thesis. Many thanks to my colleagues at the AWRI for their time on sensory panels and assistance with chemical analyses. Also to the ISVV for allowing my visit, donation of French Viognier wines, staff being on sensory panels, and especially Professor Philippe Darrier and Takis for their time, care and interest. Of course, we couldn't do this without the Australian wine industry, wine donations, enthusiastic discussions, and especially to Yalumba for access to the Viognier clonal vineyard block. Thanks to our funding body, Wine Australia, and the Australian Government Research Training Program Scholarship through UniSA for my PhD, plus the UniSA IPI funding and the BAG Alliance enabled my visits to the ISVV in Bordeaux and several international conferences. Thank you.